All right, hi again, everybody. Now we're going to talk about some of the additional special senses. So in this lecture, we'll talk about taste and olfaction. In the next lecture, we'll talk about hearing and equilibrium. So this is an area of the brain that, or rather the cranium that you're familiar with. The organ of smell is this yellow tinged patch over here. And it's about 5 centimeters squared of pseudostratified ciliated epithelium, and this is in particular referred to as the olfactory epithelium. That's the organ of smell. So basically, if we're down here, this is where the air is coming in from your nostrils into the nasal cavity, and the air is passing through underneath this olfactory epithelium before it reaches your respiratory tract. The olfactory epithelium contains millions of these olfactory receptor cells, which have non-motile cilia that are at the ends of the dendrites. So there's the little cilia right there. And that's where the sort of sensor proteins are found, the receptors for this. The reason cilia are there is they're good antennae. They decrease or increase the surface area to volume ratio. They're surrounded, these olfactory receptor cells, by a number of support cells. These are columnar cells that secrete mucus, and in fact this blue mucus that the cilia are embedded in and helps them identify their stimulus are created by those supporting cells. On the basal surface, there is an additional population of cells which are in blue here. There's one. There's called basal cells. And these are the stem cells for the epithelium. So you could actually, in theory, damage your olfactory epithelium and it could regenerate itself based on the divisions of these basal cells. The axons of each of the olfactory receptor cells are bundled into filaments of the olfactory nerve. So here you can see them, these axons now bundling together, and then they're passing through these plates, these little gaps in the bone. This region of the bone is called the ethmoid bone, and the point in which there are these pores in the ethmoid bone that allow these axons to bundle through and reach ultimately their target cells up here. This is called the cribriform plate of the ethmoid bone. And so you can see the filaments of the olfactory nerves ascending beyond or superior to the ethmoid bone. Those axons then synapse with the mitral cells that are found in the olfactory bulb. So this yellow thing right here is the olfactory bulb. It's the end of the olfactory nerve. It's just the tip of the olfactory nerve. The mitral cells then project their axons. You can see they're all now bundling together into this olfactory tract. And if we followed this would lead to the olfactory nerve. So that's your organ of smell, and you can see that right now we have at least this cell is responsible for detecting the stimulus and then sending it to the second cell, which are these mitral cells, which will then send this out through the olfactory nerve, and then it will reach the appropriate region of the cerebrum. Oops. This pathway that's responsible for the detection of smell is actually very similar to the pathway used for the detection of vision. So we have, again, a G protein coupled receptor right here, which is linked to a G protein. If the signal, in this case, which is an odorant molecule, binds to the G protein receptor, it frees up the G protein to go across the membrane, bind to an enzyme, which will spike some sort of second messenger, which will then change the behavior of this channel here, and ions are going to do something. Okay, so first step is the odorant binds the receptor. The receptor now can activate the G protein, so the G protein diffuses across the membrane to bind to this enzyme called adenylate cyclase. And adenylate cyclase takes ATP and converts it into this second messenger right here called cyclic AMP. Cyclic AMP is what gates the channel, and so when you have a spike in the second messenger cyclic AMP, it will bind to the channel, open it up, and it's a cation channel, just like in the photoreceptor story. But now in the presence of the signal, the channel is opened, it's a cation channel, so sodium and calcium rush in, the cell becomes more positively charged, which is a depolarization, which will get you closer to action potential. So notice that this is doing the opposite of the original phototransductor cells in the vision pathway. So in the presence of the stimulus here, the outer end, we get a depolarization and an action potential. Remember when we talked about the visual activation of photoreceptor cells, the light activating the G protein caused the turning off of the cation channel and hence the lack of action potential. So in light, you are not making action potentials. In dark, you are making action potentials. In the smell pathway, in the presence of the smell, you are making action potentials just like you would expect. Okay. The next organ we want to look at are taste buds. Those are the sensory organs for taste. We have about 10,000 of them, and most of them are on the tongue, although there's a few on the cheeks. There's some on the pharynx and in the epiglottis, so you can taste there as well. Most are found in these papillae, which are these small projections popping up on the surface of the tongue. There are fungiform papillae, which represent the location of most taste buds and are scattered along the tongue's surface. There are foliate papillae off to the sides, which appear as these folds at the sides of the inferior tongue. And then there are, which are not included in the original figure, circumvallate papillae, which are the largest papillae but are the least numerous. And you can see they're kind of stacked towards the posterior of the tongue there. If we zoom in on the taste bud, what we see is something like this. So there's a population of cells here with really two main cell types. The first cell and the cell that's responsible for detection of taste are these gustatory cells. And the second population of cells are these basal cells right here. And the basal cells are the stem cells for gustatory cells. And so they're incredibly regenerative. And you know this because all of us have accidentally, for example, I'm having coffee right now. And if the coffee was too hot, I could burn myself. And everybody knows that feeling of burning your taste buds. But you also know that your taste comes back pretty quickly. So these have an incredibly regenerative property. Gustatory cells, if we zoom in on them, have these little gustatory hairs that are projecting out into sort of the opening of the tongue that the taste bud emerges. These microvilli extend through this region here, which is called the taste pore. And you can see actually the tongue itself is a stratified squamous epithelium, which is what you'd expect. Unlike the other examples, these are actin-based structures. They are not cilia. And then finally, you can see, if we look at the back of the structure, you can see bundled around the gustatory cells are various dendrites of cranial nerves, axons of cranial nerves. And so you should actually know which are receiving axons from this taste organ, which cranial nerves there are, right? So there's the facial nerve, which takes senses for the taste of, I think, the first 2/3 of the tongue, the glossopharyngeal nerve, which covers the posterior 1/3 of the tongue, and then I believe the vagus nerve gives you some detection of taste in your epiglottis as well. I just want to, before I go into the specifics of taste, just say the limitations of senses are a really remarkable story. So for vision, human beings are just able to detect the what we call the visible spectrum between 400 and 700 nanometers of light. But there are things that are beyond that or below that, like ultraviolet and infrared, that other animals can see, but we can't due to the specific limitations of our system. In terms of olfaction, there's about 10,000 unique odorant molecules that we're capable of detecting. And if you were considering a dog or a cat, might be considering something more like 1 million unique odorant molecules. Dogs in particular can smell like a landscape, the way we would see a landscape, they can smell it. In terms of hearing, there's this various frequencies that you can hear. Even the low frequencies that you can't hear, you can technically detect through vibration as well. With taste, it's much, much more restricted. There are 5 tastes: sweet, sour, salty, bitter, and umami. I was actually in grad school when they discovered, or I actually met the guy that discovered umami from Japan. Sorry for the name drop. So these tastes are representative of the molecules that you'll find in the food. So sweet, for example, of course, comes from various sugar molecules. Sour is coming from acids. Salty is coming from metal ions, like sodium chloride, for example. Bitter is alkaloids, so this could be caffeine or aspirin. If you've ever bitten down on an aspirin tablet, that's a very bitter flavor. And umami is the flavor of meat. It's the savory flavor, and so usually it's resulting from your taste buds responding to the amino acids in the meat. You can actually mimic this umami flavor with something called monosodium glutamate, or MSG, which is not quite as in favor in restaurants anymore, but restaurants used to add it to food quite a bit because it made the person eating the food feel like they were getting more meat than they actually were. And since meat are usually the expensive items in a dish, this can cheaply make a dish seem like it is more filling than it actually is from a protein standpoint. Just to show you how taste transduction takes place, in the case of salty and sour, it's really quite easy, because remember, salty basically means you have an excess of sodium ions, and sour means you have an excess of protons. So really what you can do here is, for salty, sodium, if you have a whole bunch of sodium out here in your food, there's nothing to stop you in these taste buds from having sodium channels, and then the sodium will do what it always does, which is diffuse into the cell, causing a depolarization, which then can result in neurotransmitter release and the information getting sent to the next neuron. So that's pretty simple. Simply, you have an excess of salt in your mouth that will able to be diffused through the sodium channels in the taste buds, and that in of itself creates a depolarization. Same thing with the proton. If we have an excess of protons because we just had something very sour, we have proton channels in the membrane of the taste receptor, and so the protons simply diffuse into the cell, and again that causes this depolarization of the membrane, which results in neurotransmitter being released into the next cell. For the other three, bitter, sweet, and umami, we actually have more of a G protein pathway again. So if we looked at those microvilli, we would see, again, there's a G protein receptor here. There's a G protein receptor here. It binds to what's called the taste-it molecules. In this case, it would be alkaloids, sugars, or amino acids. That will open up this gusteucin protein, the G protein, to diffuse across the membrane and bind to an enzyme. The enzyme in this case is called phospholipase C, which creates a second messenger that you, of course, don't need to know, called inositol triphosphate. And ultimately, the result of that is to open up intracellular calcium stores, so the calcium diffuses out of these calcium stores, enters into the cell cytoplasm, which will cause a depolarization event, which will lead to an action potential and will cause the signal for the taste to be perceived. Okay, so that covers taste and smell. And in the next lecture, we'll talk about hearing and equilibrium. Take care.